Hey everyone, and welcome! Today, we're diving into a really cool technique that lets us peek inside the secret world of bacteria. So how can we actually see what bacteria save for a rainy day? I mean, it's a great question, right? Think of it like a tiny pantry or maybe a savings account. They're stocking up on energy for when times get tough. But actually seeing those tiny stockpiles, well, that's the real trick. And that's exactly what we're going to break down. All right, let's start with the basics, the staining principle. Before we do anything, we've got to know exactly what it is we're looking for inside that cell and why our stain is the right one for the job. And what we're looking for is this stuff, polybeta-hydroxybutyrate. Yeah, it's a mouthful, so we're just going to call it PHB. You can basically think of PHB as little energy bars that the cell stashes away for later. They're just these granules of stored carbon tucked away right inside the cell. Okay, and here's where it gets really clever. These PHB granules are hydrophobic. It's just a sciencey way of saying they hate water. They're basically like little droplets of oil. Now, the rest of the cell, the cytoplasm, that's almost all water. So you've got this classic oil and water situation going on. And that, right there, is the secret to this whole technique. So if our target is oily, what do we use to find it? Well, you need something that loves oil, right? That is where our special dye comes in, Sudan Black B. It's what we call lipophilic, lipid-loving. It's basically designed to hunt down those oily PHB granules and completely ignore all the watery stuff around them. Okay, so we know what we're looking for, and we've got the perfect dye for the job. Now for the fun part, the mechanism. Let's take a look at how the magic actually happens. It's a really elegant three-step process. First, the Sudan Black B dye gets inside the cell. Then, because it loves lipids, it makes a beeline right for those fatty PHB granules, dissolving into them and staining them a really deep dark color. Finally, just so we can see the rest of the cell for context, we add a counterstain like safranin, which gives the cytoplasm a nice light color. And what you get is this, a perfect view under the microscope. You get these incredibly distinct, darkly stained PHB granules that just pop against that lightly stained background. The contrast is fantastic. You absolutely can't miss them. All right, so this looks really cool, but so what? Why does this actually matter in the real world? This is where we get to the applications. And trust me, this is way more than just making pretty pictures. For starters, this stain is a super quick way to see which bacteria are PHB producers, which helps us tell different species apart. It's also an awesome tool for studying how bacteria manage their carbon and energy, their whole metabolic game plan. But honestly, that last point on the list, that's the real game changer. Get this, those tiny microscopic granules we've been staining are a key ingredient for creating biodegradable bioplastics. So by using this simple staining technique to find the bacteria that are the most efficient PHB producers, we are directly helping the research to tackle plastic pollution. We're going from microscopic granules to a massive real-world solution,